Hello, BISC1323. This is the beginning of recorded lecture 4.5, sensory systems. We got into this in the previous lecture, talking about some different types of receptors, but now let's get into the actual senses, starting with somatosensation. So this is just a fancy word for touch. Our sense of touch employs, and this is one of the things that we defined last time, mechanoreceptors, obviously responding to force and pressure. Makes sense that our sense of touch would use these things. Here we can see a cross-section of skin. Oh, there's the epidermis, the outer layer of skin we talked about a couple chapters ago. But it's down here that we see these sensory receptors. Let's zoom in a little bit further, and we can see there are actually several different types of receptors here. But you don't need to know the names of all of these because it's, I think, beyond the scope of an intro course to talk about the difference between a Merkel's disc and a Ruffini ending. What I want you to know is that within these mechanoreceptors that are responsible for our sense of touch, there are several types. They do vary in where they are found on the body and in what specific stimuli activate them. Again, some are only on the lips or the palms or the soles of the feet, and some are only in the parts of the body with hair, and some of them respond better to light touch or shapes or edges. Again, all that detail is not something we worry about. Just take in this statement that they vary where they're found and the specific type of touch that they are activated by. So our sense of touch obviously uses mechanoreceptors, but our sense of touch uses other receptors as well. We can feel, force and pressure. We can also feel temperature. We can feel how, hot or cold something is by our sense of touch. And indeed, these are thermoreceptors. They're supposed to be stimulated by temperature and changes in temperature, but many of these thermoreceptors can be tricked by chemicals. That doesn't make them chemoreceptors because it's not their job to be stimulated by chemicals, but some of these thermoreceptors can indeed be stimulated by chemicals as well. What do I mean by this? Well, there are actually a couple of very common examples of this. One of these chemicals is something called capsaicin. There's the chemical structure, molecular structure, if you're curious. Thank you, Google image search. But if you've ever had a really spicy pepper, your mouth feels like it's on fire. It's not actually any warmer. It's not actually creating any heat. It's tricking these thermoreceptors. And we think of this as something in our mouth because it comes from things that we eat. But look at this arthritis pain relief. Capsaicin, 0.1%. That's the active ingredient. So these thermoreceptors are located not just in the mouth, tricked by this chemical. Capsaicin in hot peppers, it feels like heat. And of course, I have to bring up the opposite of heat. There are also chemicals that can gum commercials and breath mint commercials make you feel like you're colder, like your mouth is colder. And yeah, this is menthol from mint oils. And yeah, it looks like you can rub this on your skin as well to feel that cooling sensation. Again, it's tricking these thermoreceptors. So menthol is found in mint oils and it feels cold. Now, another thing that we can sense with our sense of touch is we can sense when things are too much touch, when there's too much force, when there's too much pressure, or when things are too hot or too cold. We can sense pain. Now, this is actually not part of our sense of touch. This is kind of like a bonus sixth sense. Our ability to sense pain is associated with our other senses, but really, it's a separate thing. This category of receptors called nociceptors are receptors that sense pain. And again, nociceptor is an umbrella category because there are mechanoreceptors that sense pain, there are thermoreceptors that sense pain, and there are chemoreceptors that sense pain. Again, this is not part of our sense of touch, but I'm bringing it up here because it makes sense to bring it up here. But yeah, this is kind of like a sixth sense, our ability to sense pain. Okay. Moving on from this, let's talk about taste and smell. So the fancy name for taste is gustation. The fancy name for smell is olfaction. I'm lumping these two together because they are a little bit different, but they're mostly the same. Our sense of taste and smell both employ chemoreceptors. These can be located in taste buds on the tongue. Yep, here we see these pores with these receptor neurons, or they can be located here in the upper nasal passage here, connected to the brain. But either way, they're classic chemoreceptors being stimulated by chemicals. And it is, again, not much else to say about this. Chemicals are what we sense in our taste and smell. What I want to point out, though, is there are definitely other animals. This chapter has been kind of human focused here, but I want to point out that there are other animals that have either more of these receptors, just a higher density of them, or different types of receptors. Again, the chemicals that stimulate these receptors, that's a very specific chemical and a very specific receptor. If you don't have a receptor for a particular chemical, you're not going to be able to sense that chemical. But there are lots of things out there that other animals have receptors to, that they can smell or taste that we can't perceive at all. And so they can have more sensitivity by having a higher density of these receptors, or they can just have a broader repertoire of receptors that we don't have, and they can taste and smell things that we cannot taste and smell. But yeah, at the end of the day, classic chemoreceptors. All right, so something a little more complicated than that is going to be our sense of hearing. So before we talk about hearing, also known as auditory perception, let's talk about waves. Let's talk about what sound actually is. So sounds are what we perceive through our auditory perception. Sounds are waves of vibrations traveling through a medium. So when I say a medium, that's being kind of vague, but air is a medium and sound can travel through the air. But water is also a medium. Sound waves can travel through water. And solids are also a medium. Sound waves can go through solids as well. So yeah, that's why it's so generic. It could pass through any of those things. Sound waves, like other waves, have some basic wave properties, including amplitude. Amplitude is how high up this wave is, how tall it is, or how short it is. When we're talking about sound, amplitude corresponds to volume. Waves with a low amplitude would be quieter sounds. Waves with a higher amplitude would be louder sounds. Another property that all waves have is frequency and wavelength. Frequency and wavelength, when it comes to sound waves, correspond to pitch. A high frequency or a short wavelength corresponds to a high-pitched sound, and a low frequency or a longer wavelength corresponds to a lower-pitched sound. So there's a lot of nuance we can get from just waves. So here's the summary. Wave amplitude corresponds to volume, wave frequency and wavelength correspond to pitch. So let's look at the actual process of perceiving these sound waves. So this one's complex enough. I'm going to break it down to, steps 1, 2, 3, 4, whatever. I'm never going to ask a test question. What's step number 4? The numbering is just to sort of walk us through this. So this begins with sound waves being channeled through the outer or external ear through a structure called the ear canal. So here's a look at this process. Looks like a tuning fork creating a sound wave, these vibrations in the air at this point. And if you look at them, they have a certain wavelength, they have a certain amplitude. But here's the ear canal, and we see these waves, these vibrations being channeled in. This is the ear canal or the external ear. Here's another figure that shows it, I think, a little more clearly. Yep, ear canal, that's the external part of the ear. Once it's in the ear, these vibrations hit a structure called the tympanic membrane and cause the movement in 3 small bones, the incus, the malleus, and the stapes. You don't need to know those, just know 3 small bones. But here we see, there's the ear canal, here is the tympanic membrane, and yep, there's the malleus, there's the incus, there's the stapes. For any trivia lovers out there, the stapes has the distinction of being the smallest bone in the human body. So yeah, but these are bones. These are solid structures. So we're going from vibrations in the air now to vibrations in these solid bones. once these vibrations are in the three bones, these bones vibrate against part of the inner ear called the oval window, leading to the inner ear in this bony cochlea structure. Back to our figure here. Here's where the stapes touches this window, and this snail-looking spiral structure here is called the cochlea. So yeah, the three bones were part of this middle ear. The ear canal was part of the outer or external ear, but now we're talking about the cochlea. We're in what's referred to as the inner ear. So inside of this snail spiral looking cochlea, there is fluid, cochlear fluid. And so now these vibrations are vibrations in fluid. They cause pressure waves in this fluid, which bend structures called stereocilia present on hair cells. So what I'm going to do in the next slide is we're going to look at this cochlea, but we're going to rotate it to face you, and we're going to slice it open. So we're looking at kind of a cross section of this cochlea structure. And it looks like this. So yeah, this is a bony structure. We can see the bone around here, but we can see different compartments within this cochlea, and yeah, these would be filled with cochlear fluid. And if we zoom in even further, we can see, yep, there are the hair cells. These are sensory receptor cells, and we see these stereocilia sticking up from them. So when this fluid vibrates, that causes these stereocilia to bend. Let me zoom in on that even further. So yeah, vibrations in this fluid cause bending in these stereocilia, and it's that physical movement of these stereocilia that cause these hair cells to begin an action potential and relay that action potential to the brain for interpretation. So if you're keeping track of the different types, that makes these things mechanoreceptors. They're responding to force, to pressure. So our sense of hearing uses the same types of receptors that our sense of touch uses, which is a weird thing to think about, but definitely true. So we have this bending of the stereocilia that leads to depolarization in these hair cells. Remember, these hair cells are sensory receptors, specifically mechanoreceptors. That starts it out. Now, the reason why we are able to perceive the difference between a high amplitude wave, a loud sound, or a high frequency wave or a low frequency wave, different pitches of sound, is because this cochlea is a pretty big structure. There are lots of stereocilia located, throughout this at different layers, at different parts of this. They're not all stimulated by every single sound wave. Different regions of this inner ear are moved to different degrees by sound waves of different frequencies. So that is what allows us to perceive different types of sound waves instead of just like noise, not noise. Now, let's look at another bonus sense. So the vestibular system, as it's called, is an extension of the cochlea. It's part of this larger structure here. But the vestibular system, vibrations and sound waves are not going to make it to this vestibular system. But there are still sensory receptor cells in this vestibular system. What they are responding to is, again, not sound waves. What they respond to is the force of gravity, the force of acceleration, the force of deceleration. So this vestibular labyrinth, as it's called, because it kind of looks like, a maze of loops, I guess. The vestibular labyrinth, which is part of the inner ear, contains more hair cells, but they respond to the force of gravity, acceleration, deceleration. So this helps give us a sense of balance. Even if your eyes are closed on like a dark roller coaster or something like that, you can sense when your body is moving or when your body stops moving or when force of gravity is applied upon you. It's because of these hair cells in this vestibular system. Again, not really our sense of hearing, kind of a fun bonus. All right. There's one more sense to talk about. This is kind of a quick chapter, and that's our sense of vision. also known as visual perception. So once again, a little bit of background about what we're actually perceiving. What we're perceiving here is light, and light is a type of electromagnetic radiation. Maybe you have seen a figure like this before. So the difference between radio waves and microwaves and gamma rays and the light that we can actually perceive with our eyes is one of wavelength. And so different wavelengths of electromagnetic spectrum correspond to different types of electromagnetic radiation. So different types of this electromagnetic radiation have different wavelengths. What we call visible light is just one narrow little piece of that. Okay, so how do we actually perceive this light? Well, going to look at some eyeball anatomy. Not too much of this, but some important parts. Light begins by passing through what's called the cornea. This focuses the light, but the cornea has a fixed shape. We'll see what that means in a couple of minutes. We'll look at something else that focuses light that has a variable shape, but the cornea is fixed in shape. It's kind of like something that focuses light coming in. And yep, it's labeled right here, this outer part of the eye. After the cornea, there is the pupil. So light passes into this black dot here and further into the eye. So yeah, cornea to pupil. This allows light through. Now the size of the pupil is adjusted by the iris. So we can kind of see this here, the pupil being in the center, the iris being this green thing around it. And yeah, here's a human eye of various dilation sizes. This pupil changes in a high light environment, in a very bright environment. You don't need to let in a whole lot of light, but in a very dark environment, in order to see anything, you want to let in much, much, much more light. So this shape is, or this size is dynamically changed. So the pupil allows light through, the size of which is adjusted by the iris which surrounds it. After that, we have the lens. So the lens also focuses light like the cornea, but unlike the cornea, the lens can change its shape. Notice these little ligaments here and these muscles attached to the lens. They can contract and stretch out this shape and change the shape of the lens to dynamically change how much focus we're getting. So the lens focuses light, and yes, the shape is adjusted. If you want to know what it feels like to flex this muscle right here to change the shape of your lens, simply look at something really close up. Maybe if you have a pencil or a pen or a highlighter or something nearby, hold that up to your face really close, and you could focus on it as close as you could get it. Everything in the background is going to look blurry. But then when you focus, on the screen here and keep that pen or pencil or whatever close to your face, now the name of that pencil is gonna look blurry as you focus on something that's further away. So when you adjust to looking at something close versus far away, that is you adjusting the shape of this lens. And finally, after light makes it past this lens, it gets down to the retina. That's all the stuff in here, where we see some receptors. Retina, this yellow thing here, which is going to start action potential. So the retina is the part that houses our photoreceptors. Now, when it comes to photoreceptors, again, these are the sensory neurons that are going to start an action potential. There are a bunch of them, and they come in two different types. So there are many photoreceptors in each eye. The two types of photoreceptors are called rod cells and cone cells. They're named for their shape, or I guess the shape of this outer segment, this boxy rectangle. I guess you could call this a rod. And this one, which is sort of tapered at the end, looking more like a triangle. Yeah, I could see that being called a cone. So that's where they get their name. But the differences are not just in their shape. The differences come in what kinds of pigments they have. So let's focus on these rod cells first. These rod cells, their outer segment has lots and lots and lots of pigment. Again, it's much bigger than that of a cone cell, so these are very sensitive. If you want to know what's actually going on with these pigments, It's a complicated chemical reaction that I think is beyond the scope of this course, but there are chemical changes that are happening in response to these photons coming in and hitting these pigment molecules. That's what's starting the action potential. This chemical reaction brought upon by a photon. So these rod cells, because their outer segment is so big, because they have so many pigment discs, they're very sensitive. That makes them very effective in low light conditions. However, rod cells are not capable of distinguishing between different colors of light, so they only give us black and white distinction. In contrast, the cone cells, less sensitive, but there are different pigments in the cone cells that allow us to distinguish different colors. So cone cells have 3 types with different photopigments corresponding to 3 different colors: red, blue, and green. So curiously, what this means is because our color distinction is not particularly sensitive and our black and white distinction is much more sensitive, if you're ever trying to, I don't know, dig through your laundry basket in the low light of the early morning trying to find the green shirt, you might have a hard time telling the difference between the green shirt and the blue shirt and the purple shirt or whatever, because when you're operating under low light, you don't really perceive color as well. So anyway, rod cells and cone cells both are the types of photoreceptors here. And again, when they're stimulated, they're going to send their action potentials to the brain via the optic nerve, and then the brain is going to interpret what these signals mean. Okay, this brings us to the end of our sensory systems chapter, but again, this was kind of a short one, so I want to move on to the next chapter, which is also the musculoskeletal system. Now, at first glance, this may look like it should be 2 separate chapters, one about muscles and one about skeletons. But as we look at the function of these things, we're going to see they operate together. You really need both of them working together to have effective movement. So let's start by talking about the skeletal half of things. And this is just review types of skeletal systems. We saw lots of hydrostatic skeletons when we were, in the invertebrates chapter. Just to remind you, the key terms define this as a skeleton that consists of aqueous fluid held under pressure in a closed body compartment. So yeah, lots of invertebrates used this. We also saw exoskeletons in the invertebrate chapter. The key terms defines this as a secreted cellular product external skeleton that consists of a hard encasement on the surface of an organism. Yeah, a skeleton on the exterior, on the outside. And you know and love the third one, the endoskeleton. I want to pause and appreciate this. This is from the textbook. I didn't grab this from some weird website. This is the textbook's example of an endoskeleton. Anyway, the endoskeleton is defined in the key terms as a skeleton of living cells that produces a hard mineralized tissue located within the soft tissue of organisms. So all of these things provide similar role or fulfill similar roles. The role of a skeletal system of any type is to support the weight of the animal, protect the internal organs, and or provide an attachment point for muscles. And this is why the chapter is called musculoskeletal system. Muscles just by themselves floating around in an animal contracting aren't going to do anything unless they're attached to a tough part of the body that can be moved when the muscle contracts. And yeah, all 3 of these in different ways and to different degrees of success fulfill these functions, especially the attachment point for muscles. Now, that being said, I do want to point out that the human skeletal system does a couple of other cool things. In addition to this, the human skeletal system also stores certain minerals and produces blood cells. More on blood cells in a later chapter, but yeah, I want to note that the human skeletal system does these things. Okay, So, if we're going to focus on vertebrates in this chapter, our skeletal system has lots of bones. So, we're going to, nope, not learn all the bones of the skull. Nope, not learn all the bones of the vertebrae. No, no, now we're in French. Nine, now we're in German. Niet, yay. We're not going to memorize all of these things. Again, if you take a human A&P class, you'll probably learn all of this stuff. It is important if you're in a major or degree program that requires human A&P, but for a broad introductory biology course, I don't think we get much out of trying to memorize all these different bones. So what I do want you to note from this is, we've got bones, obviously. We also have joints, and I just want to define what a joint is. Things we call joints are things that exist where bones meet and where bones articulate against one another. And there are many different types, giving us many different types of movement. But again, all the details are not something I want to cover in this course. So, man, if that's all we're saying about bones and joints, that is making for a pretty short chapter here. We just have to talk about the other half of the musculoskeletal system, the muscle side of things. So to talk about muscle contraction, let's look at an individual muscle fiber. This is a single cell, which is part of muscle tissue. If we look more closely at a muscle fiber cell, we will see that it contains many of these myofibrils. If we zoom in on a single myofibril, we will see there are two types of filaments in this fibril. There are thin filaments here in blue and thick filaments here in red. If we zoom in even further, we'll see what these filaments are made of. But first, let me make this statement. Muscle fiber cells contain many myofibrils. Myofibrils contain two types of filaments. Let's start by looking at the thin filament, also known as the actin filament. It's called the actin filament because, well, it's made of a protein called actin. Each one of these balls is a separate actin protein, and just repeat it over and over and over again. Lots of actin come together to form this actin filament or thin filament. So thin filaments are composed primarily of these globular actin proteins. I say globular because I know this is a cartoon drawing, but the basic shape of an actin protein, like most proteins, is vaguely spherical or globular. I'm pointing out the shape of the protein because this is in sharp contrast with the protein that makes up the other filament, the thick filament, also known as the myosin filament. So the myosin filament is composed of myosin. That's a protein, hence the name. And myosin, unlike most proteins, is not globular. It's not like a sphere. It's this golf club looking thing with a elongated rod segment, and then there is a globular head. So a bunch of these myosin proteins come together to form this thick filament. And it's going to be the action of the two of these together against one another that is going to produce muscle contraction. But first, let me make this statement. Thick filaments composed of myosin proteins, and the myosin has an elongated shape with a globular head. So, okay. Now let's go through the process of muscle contraction. This is going to begin with an action potential. Okay, that's going back a couple of chapters, but our muscles are only going to contract when they're told to contract. A neuron has to tell them that they're supposed to do this. So here's a neuron releasing neurotransmitters floating across the synapse or synaptic cleft. And when they reach this muscle cell, what they are going to do is start an action potential that leads to the release of calcium ions. What does the calcium do? Well, let me get to this first. So once again, I'm breaking this down 1, 2, 3, 4, 5, 6, 7. These numbers are just to keep track of things. So in the process of muscle contraction, the neurons that are connected to muscles are called motor neurons. A motor neuron releases a neurotransmitter, starts an action potential in the muscle cell. That leads to calcium ions being released. So what did the calcium do? Well, the calcium comes in and binds to this protein called troponin. Troponin is part of the thin filament. So the thin filament is only mostly actin. There are a couple of other proteins present on the thin filament, including this troponin protein. So when the calcium binds to this troponin protein, that exposes a myosin binding site present on this actin protein. So before calcium, the binding site was not exposed, and that's why myosin was not bound to actin. But now that troponin has calcium, it moves out of the way a little bit. Now this is available for myosin to bind to. So calcium binds to troponin, a protein on the thin filament, and exposes a myosin binding site on actin. And yeah, you could guess what happens next. The myosin head binds to its binding site on the actin protein in the actin or thin filament. So myosin head binds to actin, forming what's called a cross bridge. The cross bridge is just the name for this connection between the two. Note, at this point, myosin has ADP bound. And so remember, this is the spent form of ATP. This is the low energy form of ATP. We're going to see where this comes from in a little bit, but I want to note that at this point, when the cross bridge is made, it has this low energy molecule bound to it. Okay, next we have what's called the power stroke. This is where the myosin protein sort of flexes, it sort of moves, and that is going to move the entire thin filament with it. And so note how this portion of the thin filament was up here. After the flex, the whole thing has sort of slid down in this direction. This is a very, very small movement in the grand scheme of things, but we're going to be going through this process many, many, many times to produce the overall muscle contraction that you can see with the naked eye, that you could, moves your body. So the power stroke is when the myosin head bends and moves the entire thin filament. As this is happening, the ADP that was bound to the myosin head is released. Also, the thick filament does not move. It's just this portion of the protein that moves. The entire thin filament moves. The thick filament doesn't really. So now that the ADP has been released, there's an open pocket here on the myosin head. So what happens next is ATP is going to come in, bind to that pocket, and that is what is going to detach this myosin head from where it was attached to this actin protein. So oddly enough, the binding of ATP to myosin is what causes this cross bridge to break apart. Now the ATP is going to be spent into ADP, and it doesn't seem like it's doing anything as it goes from ATP to ADP, but what it's doing is it's charging up this myosin protein. This says the cocked position, like this is a revolver or something ready to go off, but it's energizing it, making it ready to go to repeat this whole process over again. So ATP goes to ADP, priming this myosin for the next round. And indeed, we're going to go back and repeat this again. Now that it's primed, it's going to bind to another actin protein on the actin filament, make your cross bridge, have your power stroke, release the ADP, ATP comes in, breaks the cross bridge, return to the ready position, then it's going to attach to a different actin protein and repeat these steps. These, I guess they were 4 through 7 if you were numbering them like I did. Repeat these many, 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 many times. And it's the combined contraction of many of these myofibrils that results in overall muscle contraction. Again, if we look at muscle contraction like this, our thick filaments in red here, our thin filaments in blue here, many, many, many short little power strokes are going to move these thin filaments closer to one another, and that is what's going to cause overall muscle contraction. Okay, and yeah, great figure to see this process visually if you're trying to, go through the steps and are getting kind of lost. Great figure to actually see what's going on. All right. That brings us to the end of this chapter and to the end of lecture 4.5 and the cutoff for exam number 4. So we've got more systems, we've got more anatomy and physiology, but that'll be on the 5th exam. Here's our cutoff for exam number 4.